also control the growth of microorganisms using heat. We have various test methods that can determine either the time at a set temperature that's needed to kill microorganisms, or how many microorganisms are killed within a specific time and temperature. There's all types of variations of testing the effectiveness of heat in killing bacteria, especially. The TDP, or thermal death point, is the temperature that kills all bacteria in a culture of either E. coli or Staphylococcus aureus. These are specific growth cultures that are used in the TDP. So we have a set 24-hour culture of a bacteria. We've inoculated a flask, let it grow for 24 hours at a neutral pH, and then exposed it to varying temperatures for 10 minutes and determined what temperature is needed to kill that culture within 10 minutes. That would be the thermal death point. So we have a set time and we have a set concentration of bacteria. It's just the temperature will vary depending upon what the bacteria is and how dense the growth is in a 24-hour culture. The thermal death time is simply the time needed to kill a 24-hour culture at a neutral pH at any specific temperature. So here we're varying time and temperature. It could be that we have our 24-hour culture and within 3 or 4 minutes at a very high heat, the organisms are all dead. Or it could be that if we heat our culture at a very low temperature for a very long period of time, maybe 2 or 3 hours, we still get effective killing. So with the thermal death time, we vary both time and temperature on our 24-hour neutral pH culture. The DRT, or decimal reduction time, is not looking for 100% killing. Instead, we're looking to reduce the population of our 24-hour bacterial culture to 90% reduction of viable cells. Typically, if we can reduce the number of viable cells significantly to 90% of what we originally started with, then it's not too difficult to eliminate the remaining 10%. So we would take our 24-hour culture at a neutral pH of bacteria, we'd expose it to varying temperatures, and determine what temperature would be needed to reduce that population to essentially only 10% alive or 10% viable after that period of time. And notice, the hotter that you heat this culture, for example on this graph, when we heated the culture to 70 degrees Celsius, it only took a very short period of time to kill 90% of the population. In comparison, if we're simply heating that culture to 50%, notice it doesn't just double the amount of time needed, it actually greatly increases the length of time needed to kill and reduce that population to 90% of what they originally started with. So heat is definitely an important way of controlling, especially bacteria. There are a variety of other means and methods to control microorganisms. Usually we're often thinking bacteria, but in general microorganisms can be controlled by temperature, moisture, and varying kinds of environmental changes. If we use dry heat often takes longer to kill or inhibit the growth than moist heat. The addition of the water, or making certain that we have a moist heat, steam heat environment, is usually going to have more caloric energy available, or in other words, a higher temperature, than dry heat. It's going to take more energy to heat up our stove. If you compare how hot steam is with just trying to heat up a dry oven, you know that moisture or water vapor can contain a fair amount of caloric heat energy. In other words, the steam heat gets hotter faster than simply a dry oven. And that's part of why steam sterilization is a very effective and much faster method of sterilization than dry heat sterilization. They both still are very effective. It's just moist heat is much faster and as effective as sticking something in an oven. Refrigeration is good at controlling microbial growth, but it simply inhibits or slows down the growth. There are very few microorganisms that are outright killed in the freezer, maybe some protozoans, but if we're thinking bacteria, viruses, and fungi, those kinds of microorganisms are usually simply put in a static or slow state of growth in the refrigerator, with a few exceptions. If we have a psychrophile that actually grows better in cold temperatures, then putting them in the refrigerator actually encourages them to grow. That's, of course, the spoilage organisms that we find on our food. Freezing is just another more extreme case of cold or use of cold temperatures. And again, freezing typically simply tremendously slows down the growth or even puts the microorganism in what you would think of as a state of suspended hibernation or sort of waiting. It'll hold it over. You can freeze it. You can put it in the freezer, hold it over for months at a time, even years. It sort of stops the growth but does not kill the organism. And when we take it out of the freezer, simply restreak it onto an agar plate and it's ready to use again. Desiccation or drying can often be a very effective way of killing certain types of microorganisms. And if you dry them out completely, remember we're not only removing the water from the outer environment, we're also removing the water from inside the cells. And so desiccation can be very effective. Freeze drying is where we're simply rapidly removing the moisture from the cell and putting the organism into a very dry condition. Freeze drying, if done properly, actually can be a method of preservation or preserving, as can simply drying. So if you simply dry a piece of fruit, you'll notice that that fruit is still edible. In fact, you can also take the fruit, soak it in water, and rehydrate it so that it's useful again, more like a piece of fresh fruit from an apple, from the fruit itself. Well, drying and freeze drying or lyophilization of bacteria is a way that we preserve some of our bacterial cultures. The conditions under which we do those procedures have to be fairly tightly controlled. We can't just take a culture of bacteria, let it sit out on the kitchen table or the laboratory countertop and let it dry up and assume that if we take a little sample of that dried up material and put it on a fresh agar plate that it's going to be alive. So we usually have to add some types of preservatives, extra special chemicals that will protect the individual bacterial cells if we want to use desiccation or freeze drying, lyophilization, to preserve our bacterial cultures. Otherwise, by drying or desiccating our cultures, they often are killed by the complete removal of all the water. Kind of an interesting play on various growth conditions and environmental conditions and the control of our microorganisms. Radiation is another good way to control the growth of unwanted microorganisms. Either ionizing, which would be X-rays and gamma rays, or non-ionizing, which would be ultraviolet light, are very good methods of controlling the growth of microorganisms. Both kinds of radiation have deleterious effects on the nucleic acids and the proteins in the cells. And in fact, depending on the length of exposure, UV light, X-rays, and gamma rays can all be used to sterilize surfaces. We can even use microwaves, which are the other end of the extreme of wavelengths. Microwaves essentially cause water molecules inside of the cell to shake or vibrate. And of course, you probably all know that if you expose a cup of water to microwaves for a long enough time, you can get that water to boil. Well, the same thing happens inside the cell. If we expose those cells to microwaves for a sufficient length of time, we essentially cook them. Now, we've already mentioned dry and moist heat, refrigeration, freezing, freeze-drying, but we also need to include ultrasound or ultrasonication, filter sterilization, and osmotic pressure. Ultrasound is simply using sound waves, again, to cause internal vibration of the components inside of a cell so that they literally shake themselves apart. Now, the ultrasound wavelengths that are used in diagnostic investigation or evaluation of internal body structures is not at the rate of wavelength that's used for sonication or disruption of bacterial cell cultures. We have a wide range of sound wave energies that can be used for all types of procedures. And so the medical ultrasound investigation that's used to determine internal body structures is of a much safer and lower frequency than the sonicators that are used in disruption and essentially killing a culture of bacteria. Now, filter sterilization is simply using a very thin membrane that has tiny little pores or openings that will only allow liquid to pass through, but will trap any microorganism on the surface of that membrane. It looks very much like a little piece of plastic, but what you can't see is that little piece of plastic has tiny little holes that will only allow liquids to pass through. Now, the manufacturing companies that manufacture filters that we call membrane filters for these procedures can vary the pore size. So there are membrane filters that can be used that will filter everything except viruses, so we can purify viruses from a bacterial culture or from, let's say, a specimen from a person. So the pore size is just large enough for viruses and liquids to pass through, but not bacteria or anything else. There are membrane filters that would allow viruses and bacteria to pass through, but not any other kind of material. And then, of course, the types of membrane filters that we're interested in are the membrane filters that would essentially only allow liquids to pass through that we want and trap all and any microorganism on the surface of that membrane so that we can then remove or separate the microorganisms from our precious liquid. This membrane filtration is often used in manufacturing to make sure injectables and IV solutions are pristine and completely free of any unwanted microorganisms. Using osmotic pressure, making sure that we have, let's say, maybe a higher osmotic pressure in our solution to inhibit the growth of our microorganism, well, that's why jams and jellies work. Jams and jellies have a very high osmotic pressure, or in other words, they are hypertonic in comparison to the tonicity of the cells that we place in those sugary and salty solutions. By changing the osmotic pressure, either making it too low, which would be water, so hypotonic, or maybe a hypertonic environment where we have lots of salts. This is why a high salt solution is a very good cleansing liquid, because we can remove all of the water from the bacterial cells simply by applying that hypertonic salty solution. In either case, we're controlling or preventing the growth of our microorganism by changing essentially the osmotic pressure on its cytoplasmic membrane. Cells don't tolerate those changes very well, and so simply sticking your hands into a nice flowing source of water in the sink, if you don't even have time to scrub, simply rinsing your hands with water helps remove microorganisms by both changing the osmotic pressure and the mechanical surfaces being cleansed by the flow of the water. Here we have an illustration of a membrane filtration system. On the right-hand side is a scanning electron micrograph with the membrane filter in blue underneath this layer of cocci of bacteria. Notice the tiny little openings or pores in the membrane matrix. The bacteria are trapped on the surface, and the liquids are passing through the little openings or passageways in the membrane. Now, typically we filter sterilized materials or liquids that cannot be heated. So if we have any kind of very special antimicrobials, treatments, various kinds of chemical treatments that have to be used for patients, and we can't autoclave or heat up that liquid to kill any unwanted microorganism, then we will filter, sterilize it to make sure that there's no unwanted microorganism in that very special liquid. An autoclave is an instrument that's been used for decades to control the growth and kill microorganisms. With steam sterilization under pressure, we can, in this contained device, in this contained chamber, we can increase the temperature above boiling so that we have steam heat at 121 degrees Celsius, which is 256 degrees Fahrenheit. The standard procedure is to keep this temperature at this level for 15 minutes at 15 pounds of pressure per square inch on the inner surface of the chamber. That's the standard used to sterilize, or in other words, kill all unwanted microorganisms, or any microorganisms, even including prions and viroids, in our steam sterilizer. Now, prions are a problem because sometimes sterilization isn't as effective as it could be. So we'll concentrate on the other kinds of microorganisms that we're used to working with: the viruses, fungi, bacteria, and any kind of helminths or parasites. So steam sterilization, 15 minutes, 121 degrees Celsius for 15 pounds pressure per square inch. Standard, very effective way of sterilizing all kinds of materials, and frequently had been used in decades past to sterilize surgical instruments.